This is Manjunath here, and I think all of you are safe in your homes, and please stay in the home. And I'm going to do this first video lecture for second PUC students, mainly on the chapter sexual reproduction in flowering plants. Okay, students, already we started with this chapter before lockdown, that is sexual reproduction in flowering plants. In that chapter, we mainly covered so many points, and it is one of the important chapter in second PUC. It carries a lot of diagrams and important explanations. And in that first, we mainly know about the structure of flower. Okay, this already we studied. This is the structure of flower. What we studied in the previous classes. This is the structure. Already in first year also you studied about the structure of flower. The common parts are present here. Those are stigma, style, and the anther, and everything. They are present here. And usually the flowers, majority of the flowers are having two sexes in a single flower. Two sexes are present here, both male and female. And in that, mainly flower contains four whorls that we know commonly. Four whorls are present here. Two accessory whorls and two reproductive whorls are present here. First we named as outermost sepals are there, petals are there. Then after that, androecium and then gynoecium. These are the four important whorls in the flower. And in this chapter, they mainly given the sexual reproduction. How the sexual reproduction occurs in case of flowering plants. That is a very important point regarding this chapter. And here, this flower, structure of flower we already completed here. It's a modification of shoot. We know it's commonly. And then the definition of the flower and two important whorls in the flower. That is, one is gynoecium, another one is androecium. Androecium is a reproductive whorl, that is male reproductive whorl, and gynoecium is a female reproductive part of the flower. And is commonly we call it as androecium is called as a stamen, and then gynoecium we call it as a pistil. Okay, pistil we call it, commonly we call it as like that. And the next, this is the stamen. Already we completed this one. The male reproductive unit. It mainly contains filament and then anther. Here actually the microspores are going to be produced here. And next to this one, that is this the structure of anther. Very important diagram, and in NCERT it is different, and what I am using here it is different, but in NCERT they given the well diagram, and that you have to practice. This is the important diagram. Then next to that, structure of anther, that is nothing but microsporangia. This also I already explained, and usually it is diathecous. It means it contains two theca. Bilobed, also we call it as a bilobed. The structure they give here, the longitudinal grooves are present here, and then cross section. If you observe the cross section, it is tetragonal in structure and it contains four microsporangia. If you draw the diagram like this, it mainly contains four microsporangia. This is 1, 2, 3, 4. Four microsporangia are present here, and here later two microsporangia of each lobe fuse with the pollen sac. Okay, here they are going to be fused here. Pollen sacs. These microsporangia is commonly called as a pollen sac. These are the sac-like structures. These are already explained regarding this. Then still it is continued, and then it mainly contains the diations are present here, and usually it contains four layers. Those important four layers are present here. Those four layers are epidermis. Here you can find the epidermis, endothecium, middle layer, and then tapetum. Okay, if I draw the diagram like this, it is mainly contains four layers. Three main layers are present here. The outer one is epidermis, next to that endothecium, next to that middle layers are there, and this is tapetum. This is tapetum. pattern what they given here. Okay, this is the structure of anther, or we also call it as a microsporangia. And then next here in that anther, uh, microsporogenesis. Study that anther structure. That is very important. Please remember, for the diagram they will ask the question. Draw a neat label diagram of tails of anther, or they will also call it as a microsporangia. Draw a neat label diagram of microsporangia. Don't get confused. And here next important, that is microsporogenesis. How the microspores are going to be produced. This also already completed. Here the microspore tetrads are going to be formed. There is a presence of pollen mother cells. Those will undergo maturation and they will form the pollen grains. Pollen grains are nothing but the microspores. And the process of formation of microspores, we call it as a microsporangia. And the next important term, that is pollen grain, that is male gametophyte in case of plants, and that mainly exine and intine. Usually two layered wall. One wall we call it as the exine, another one we call it as the intine. Exine is mainly made up of sporopollenin, and it is very highly resistant organic matter, and it can withstand high temperature, acids, alkalis, enzymes, so many things. They will not degrade very easily. Okay, these are very rigid organic substance. It will not easily going to be degraded, and it is mainly made of that. And then it is important, they will ask the question for your NEET as well as for CET. They will ask the question sporopollen. Exine is mainly made up of sporopollen. And then after that there is a intine, a thin continuous layer mainly made up of cellulose and pectin. Important, please remember, exine is mainly made up of sporopollenin and intine is mainly made up of cellulose and pectin. This is important, please remember from this side. This is the pollen grain structure or male gametophyte. Okay, still few points are left here. That is, here pollen grains, and then here the mature pollen grain usually contains vegetative cell and then generative cell. Two types of cells we can find here in pollen grain. One is vegetative cell, another one is generative cell. And in vegetative cell, it mainly contains the abundant food reserved. A lot of food materials are present here. As the name itself indicate, vegetative. Vegetative, it means it provides abundant food for the development of the pollen grain. And next one, that is generative cell, is mainly from the generative cell there is a formation of that pollen tube during the fertilization that takes place in generative cell. With the help of generative cell, they are going to produce the pollen tube for the fertilization. Okay, these are the important points regarding male gametophyte or pollen grain. Okay, and this is the thing. Okay, coming to the next one, that is gametophyte, that is female gametophyte, that is pistil, or we call it as a pistil or gynoecium. Already completed the androecium. Here I given gametophyte, and that is a gynoecium. And here this is also called as a pistil. And this pistil usually contains mainly three parts, that is stigma, style and ovary. Stigma, style and ovary. If we draw the simple diagram here, this is the structure, this is the structure. Okay, this part we call it as a stigma, and this is the slender elongated structure, we call it as a style, and this is the ovary, and inside the ovary there is a presence of ovule. Okay, this is the common structure, this we already know.
And this gynoecium is a very important female reproductive part of a flower. Okay, gynoecium is a important reproductive part of a flower. This is the female reproductive part of a flower. And in last year, that is in first year, we already studied the carpellary. Okay, carpellary, pistil, all are same. But in case of morphology of flowering plant, we studied about the number of carpels. If they contain only one pistil or only one carpel, such pistils we call it as a monocarpellary, or such gynoecium we call it as a monocarpellary. And if it may contain more number of carpels or more number of pistils, those are we call it as a multicarpellary. Monocarpellary if they contain only one pistil, multicarpellary if they contain many pistils. Okay, this already we know. And along with that, another one term we know that in case of some, if it is then multicarpellary, in those cases what will happen, sometimes they are fused, and such fused condition we call it as a syncarpus. Please remember this word from the last first year. You already studied about this syncarpus. If they are fused, if they are fused, if they are free, those are apocarpus. Carpels are single in number. If they contain only one pistil, such carpels we call it as a monocarpellary. If they contain more than one carpel, or if they contain more than one pistil, those we call it as a multicarpellary. And in that only, if the multicarpellary and gynoecium, if they contain more number of carpels, sometimes they are fused or sometimes they are free. If they are fused, that is syncarpus. If they are not fused, if they are free, those we call it as a apocarpus. And usually this structure, you can see at the pistil, it mainly contains a stigma which receives the pollen grains, or it's a platform for the pollen grains. What will happen during the pollination? This is the stigma. All the pollen grains are going to be come and settle here. So it is act like a platform to receive the pollen grains. So this is the stigma. It mainly receives the pollen grains. And then next is style, which is the elongated slender part below the stigma. Here you can find this structure. We call it as a style. This is style. Elongated structure. Sometimes they are fused, bulged. Sometimes they are hollow. In some cases they are bulged, and in some cases they are hollow. Okay, for what purpose these styles are useful? Just for the transfer of these pollen tubes. When the pollen grains start germination, so these pollen tubes can travel like this, and they will reach up to the ovary. This is the importance of style. The next to that, that is ovary, which is a bulged part, the end part, or a bulged part, that is the bulged part or the end part, that is ovary. Here you can find the stigma. This elongated structure is a style, and this bulged part of a gynoecium we call it as a ovary. This is ovary. And inside the ovary there is a presence of ovule. Inside the ovary there is a presence of ovule. Ovary may contain more number of ovules. Number of ovules are present here, and these ovules are located in the locules of the ovary. There is a presence of locules. For example, if I draw the diagram like this, here these are the locules of the ovary. Like this, you can find here the locules of the ovary, and inside the locules of the ovary we will find the ovules. It means inside the ovule there is a presence of ovules, and inside the ovule there is a presence of embryo sac. And this how these ovules are arranged, they are attached to the placenta. If you think it is a placenta, here there is attachment of ovules, there is attachment of ovules. Okay, this is the structure. The placenta mainly contains the microsporangia. Placenta mainly contains the megasporangia or ovule. It means if we consider this is the placenta, here there is a presence of ovule. It is attached like this. Okay, just see here, inside the ovary there is a presence of locules. Those locules we call it as a ovarian locules, and inside the ovarian locules there is a presence of ovules. Inside that there is a presence of ovules, and inside the ovules there is a presence of embryo sac. Inside the ovules there is a presence of embryo sac. Embryo sac is going to be present inside that. Okay, understood regarding this? This is the gynoecium or pistil. Okay, very simple, already studied in the first year. The same thing, exact the same thing is present here. Again I am explaining about this, and in first class only, okay, I have explained about all these structure of androecium and then gynoecium. Again the same thing is present here, that just go through once. If you go through once continuous reading about this, you will came to know the idea. And these are all the basic for high deep concept. So for that you should know about all this. Okay, next, that is megasporangia. Next one, that is megasporangia. Megasporangium is nothing but what we call it as a, we'll call it as a ovule, not ovary, it's a ovule. Megasporangium is nothing but a ovule, and usually here they given ovule is attached to the placenta by a funicle. If you observe the diagram, I will show the diagram for this. Here in your NCERT they given the same diagram. This diagram is from NCERT only, and this is the structure, complete structure of gynoecium. Stigma here, style here, and then ovary is present here, and this is the thalamus. Okay, that is the thing. And coming to the structure of ovule, anatropus ovule, or it is also called as megasporangium. Please remember, this is the hilum. I will first tell you the parts. This is the funicle, this is microphyllum, this is microphylar pole, this is the outer integument, this is the inner integument, nucellus, embryo sac. Students, please remember this diagram is very very important, because so many times they ask the diagram. They'll ask either draw neat labeled diagram of TS of anatropus ovule. Very important, so many times they ask this diagram. Okay, please remember, please practice this diagram. That microsporangia also they ask many times, that is anther, structure of bilobe anther. And here in case of female gametophyte, they will ask 100%, they will ask the diagram of this anatropus ovule. Okay, please remember this diagram, very important and very simple. Okay, coming to the explanation regarding this diagram. Okay, this is the structure of anatropus ovule. This is the structure of anatropus ovule, and you can find different types of parts here, or different parts are present here. If you observe the structure, there is a presence of this outermost layer, that we call it as the outer integument, and next to that there is a inner integument. Okay, please remember the parts. They will mainly protect the embryo sac which is present inside here. They will protect the embryo sac. the outer integument, next to that inner integument, then next to that, after these two integuments, there is a presence of nucellus. Here they given nucellus, not nucleus. Please remember nucellus. Nucellus is a mass of cells. Those store the food material or nutrients for the developing embryo sac. Okay, nucellus is a mass of cell. Clusters of cells are present. Mass of cells are present here. Okay, and inside the nucellus there is a presence of embryo sac. Inside the nucellus there is a presence of embryo sac. Already we studied about the structure of embryo sac, and simple diagram we studied, and a further detailed structure we will study in the next upcoming classes. Please remember, this middle one is the embryo sac. This structure we call it as a embryo sac, and the embryo sac is mainly contains different types of cells. Antipodal cells are there, synapsid cells are there, polar nuclei is there, then egg cell is there. Okay, that we will study in the later. But now here the simple diagram, and along with that here there is a two ends are present here. In here there is a two ends are present. One is chalazal end, which is opposite, and upper end, that is microfill. There is a small opening. These integuments are not completely covered the nucellus or embryo sac. Okay, there is a small gap present in the upper region of the anatropus ovule. That region we call it as a microfill. And here after this, another two important parts, one is hilum, another one is funicle. This funicle is mainly attached to the placenta. Just consider this is the placenta. It's a stalk like structure, elongated stalk like structure, that is funicle. It is an elongated stalk like structure, that is funicle. And this funicle is attached with the body of the ovule. This is the body of the ovule. This body of the ovule is attached to the funicle, and these two funicle, the stalk like funicle and body of the ovule, they fuse in a common point or a common area. That area we call it as hilum. Here it is. This is the hilum. Okay, this structure we call it as a hilum. Hilum can connect the funicle and body of the ovule, anatropus ovule. Okay, this is the simple structure of anatropus ovule. Listen, once I will tell you. It is mainly contains different parts. It contains outer integument, inner integument. They mainly protect the nucellus and the embryo sac. There is a mass of cells present inside the, or after completing the two layers, outer integument and inner integument. There is a mass of cells we can find here, that is nucellus, and inside the nucellus there is presence of embryo sac. These nucellus mass of cells, and these are mainly reserve the food material or nutrient that will provide for the embryo sac. Okay, apart from this, it mainly contains two poles or two ends. The upper end we call it as a microfill, and the lower end we call it as a chalazal end. And along with that, it mainly contains the funicle. It is a stalk like structure which is attached to the placenta. It is attached to the placenta. Then this funicle and polar body, or sorry, anatropus ovule or ovule, they are attached in a point, and that region or that area is we call it as a hilum. Okay, it is a very simple chapter. I hope so. All of you understood the concept. Okay, and if you have any doubt, you can ask through WhatsApp. Okay, thank you.